Hey everybody, we're going to take a look at bacteria today, and that's part of chapter 4. So we're going to skip from chapter 1 to chapter 4. 2 and 3 we're not going to do, and therefore if you're wondering, we are skipping ahead there. So chapter 4, we're going to take a look at bacteria, and I pull up some images here to give us some visuals before we start. If you recall from chapter 1, we talked about bacteria as being one of the common types of living things. I also described them as being ubiquitous. So if I didn't make that clear, or just to recap, microbes are ubiquitous. That means they're found everywhere, but they're not all necessarily found equally. So when we say microbes, that's a blanket term. When we get to the specifics of the types, bacteria are really one of the most ubiquitous types of microbes. Other microbes, things like protozoa, algae, not nearly as common, especially just ubiquitously. They are more common in certain areas, but not throughout. Bacteria really are one of the only microbes that are truly ubiquitous. I mean, you can literally find some kind of bacteria almost anywhere you go. And that's not true for other microbes. So as a whole, they are ubiquitous, but individually, some types are more widespread than others. Bacteria is certainly one of the most widespread. Arguably, equally as widespread would be viruses. Viruses are thought to even be more quantitatively higher. In other words, there's more of them. Probably just as you can only get everywhere once, right? You can't do it on a different level. So once you're everywhere, you're everywhere. And the truth is, bacteria and viruses are thought to be everywhere simultaneously. So everything else is kind of in certain locations, but these are everywhere. And what we want to understand is what they are, a little bit about them, what are the different characteristics and types and things like that. So that's the idea. So here's some images of some bacteria of circular nature, and we'll come back to this when we talk about some of the terminology right off the bat here. So one of the problems I'm having is finding a good size comparison, and I've just really not been able to come up with something like that. So I do apologize. It's difficult to gauge the size here. I will try, but I've just not found any great resources to help me on that. So nothing that I think really does a good job of it anyway. Okay, but let's get started here, and let's just start by going through what it is we're going to talk about. So we're going to talk about the shapes, and we're going to talk about what's called the arrangement, which is a grouping pattern. We're going to talk about some of the structures they have, and that includes things on the outside. That includes kind of the way that their cell membrane, what kind of makes them. Think about it like the casing around the bacteria, kind of like their skin is a kind of way to think about it. Is the cell envelope. We're going to talk about that. And then we'll talk about some of the internal structures. Not as much, but a little bit about what's going on inside there. So that's the idea. Shapes, arrangements, and structures, and then a little bit of internal structure here. Okay, we're going to start with the basics. And what we want to start with here is just the shapes. So bacteria are these single-celled organisms, and they each have their own distinct shape. And while there are lots of different bacteria and lots of subtle variations in shape, there are really a handful of what microbiologists have classified as the most common types of shapes. So they're really more like a generalization about what types of features or shape features are most common, and that's what we're looking at here. So the three most common general shapes are referred to as coccus, bacillus, and spirillum. Now, these are Latin derivatives, and they don't necessarily have an English context, which is kind of frustrating. A lot of science, as you probably already know, is like that. So cocci, or coccus, excuse me, is a word that stands for spherical or circular, and therefore that is the context here. So spherical, circular. Now, let me pull up something here to make sure we cover this here real quick. There are two different contexts. So coccus equals a singular, and cocci is plural. So the last part there, the suffix us versus I, changes that from a singular to a plural. So if you're talking about a singular bacteria, you'd use the word coccus. If you're talking about multiple, you'd talk about cocci. That's also true for bacillus, which are rod-shaped cylindrical type cells, sometimes called a box-shaped. Bacillus, again, bacilli. If you drop the us and put an I on there, to spell it out real quick here. Bacillus and bacilli. rod shaped. So let's go back to the figures. So here's a cockeye. I pulled up that initially. Circular, somewhat spherical. Again, they're all just a little different, but that's kind of the general theme there. That is not. That's an exception to that. Bacilli here are more of a rod-shaped type cell. Okay, you can kind of see not boxy. It's not really a true boxy. They're always kind of curved, but they tend to have more of a long, stretched out cell shape to them. Okay. So coccus and bacillus. Now, truthfully, these are the two most common, really, by a significant margin. The third most common, and especially in the soil and in the water, are bacteria called spirillum. And spirillum, once again, is the singular. And spirilla is a plural. Spirillum have a spiral shape. This one actually makes sense for once. So we have a spiral shaped type cell there. Okay, so those are the three most common shapes. When you look at bacteria across the world, they typically conform to one of those three. Now, the most common, the most common variations of that, in other words, subtle, kind of a subtle mix in some cases. So, for example, a coccobacillus or coccobacilli, plural, is kind of is a mix between the two. Let's pull that up real quick here. Coccobacilli are going to be a little bit nubbier, so to speak, than the bacilli. So they are kind of a mix of the two there. All right. Vibrio is going to be a slight variation of the bacilli in that it's going to have a little bit of curvature to it. So you can see here we got a bacilli, but it's got kind of a bent shape to it. Okay, you can kind of see that here. Here's another picture. Not quite a straight rod shaped. Okay, it always reminds me of somewhat of a squash shape, and that I think of them as a squash type bacteria. And each one of these is going to be a little different, but that's the kind of the difference there is a kind of a curved rod shape, curved bacilli. And then a spirochete is a lot like a spirillum. In fact, let's see if we can do a comparison. They're more of like a corkscrew. Here's a good figure showing all of these here. But spirillum is more of a gentle curve, whereas a spirochete has more of a hard twist in it, really a lot like a corkscrew. And in fact, I'll talk about these next here in my PowerPoint. So the difference is in the kind of the curved nature itself. So I think of this as almost more like a noodle type curve, and this is more like a corkscrew type curve. So there is a significant difference there. Okay, so you want to be familiar with those. What I will do on these is give you basic descriptions that you will be asked to match or describe in the shorthand or maybe even multiple choice. What I won't do is I won't try and trick you between subtle differences in with pictures and imagery. I won't try and give you something that is somewhat hard to determine. So, for example, if we go back to the coccobacilli, those are. What I would not do is I would not give you this picture and ask you to tell me whether it's a cockeye or a bacilli. Because honestly, if you told me that was a cockeye, it'd be hard to argue because there is a slight difference there. So I won't try and trick you on that. But if I did give you that, I would point out that it's shorter or that it's got kind of a half rod shaped, half circular. So I'm not going to try and make this unfair. And so just to clarify, when I give you these questions, I will try and make sure you understand the context. So it is almost subjective at a certain point as to what it is. Okay, so just know the basics, no tricks here. If I do, you can call me on it. Okay, next we want to talk about the difference between the spirochetes and the spirillum here. I already kind of mentioned it, so actually what I want to do is kind of just clarify that here. I mentioned it, I have it in the PowerPoint. The spirochetes are often referred to as the corkscrew, quote-unquote, bacteria. Okay, not the technical term, but really commonly used way of describing them. And the reason is because they literally have that corkscrew shape, as I already mentioned. Now, one detail I want to add to this Actually, I want to add a detail and go and refer back to something we talked about in chapter 1. The flagella is referred to as an axial filament, and the flagella is like a tail. And in fact, I'm kind of putting the cart before the horse here. We're going to talk about flagella, but we haven't got there yet. But flagella is like a tail, and if you look here, there's a spirillum down here, and it's a tail that usually hangs off one or both ends sometimes. With most bacteria, it acts like a true tail hanging off the end, providing a whip-like motion. With a spirochete, that tail is actually embedded within the bacteria itself, within the membrane of the bacteria. And so what you get is a tail, rather, it's more like a twisting mechanism than it is a tail. And what ends up happening is that the bacteria can contort this tail-like flagella and actually get it to rotate its body like this, with a twisting kind of driving motion. And what that does is it allows it to move through its environment in that kind of corkscrew type motion. Now, if you refer back to chapter 1, I actually mentioned this. In fact, let's just pull it up real quick. I mentioned this along with the disease about Lyme, or the disease about Lyme disease, excuse me. The disease, Lyme disease. There's a bacteria called Borrelia burgdorferi that is a spirochete that is the cause of this disease, Lyme disease. And so just to connect this back, this is a disease caused by a spirochete. The spirochete is what we were mentioning. And the disease in this case, if you recall, is spread by ticks. It's the bacteria that's spread by ticks, but the disease itself is really caused by the bacteria twisting and corkscrewing, so to speak, through your body if you were unfortunate enough to get this disease. And a lot of the symptoms are based upon the nature of the bacteria and kind of how they maneuver through the body. Syphilis is another disease I could have mentioned here. Syphilis is caused by a spirochete. Damage is accumulated in a similar way. And there's other ones that are similar. Leptospirosis is another one that animals get frequently. I think people can get it, but rarely do. So anyway, there's a variety of diseases here, not a lot, but a handful, that are caused by a spirochete bacteria, one that can get into the body, and really a lot of the way it causes damage is simply by the way it kind of moves through the body in that corkscrew motion. That's very unique compared to other bacteria. Other bacteria don't do that. So that's the difference between those two there. This is a figure out of your text showing you some different examples of the different shapes here. They added one here, the branching filaments. I didn't mention that. It's not usually mentioned amongst the most common, and ironically, they put it in the figure but didn't really mention it themselves amongst the most common, so I excluded that one. But anyway, you can see these here. These are images I'd be likely to use on a quiz or something like that, or test. Sorry if I keep bumping the mic. I'm not sure if you can hear that. Okay. Bacteria sometimes don't have a defined shape. They don't. The cells within a population don't conform to a single shape. And when that happens, the term is used called pleomorphic. So the term pleomorphic is used to describe bacteria that don't conform to a single shape. So that's the idea here. Pleo means more than usual or many, and morphic is in reference to shape or character. This bacteria we mentioned in chapter 1 as well, Helicobacter pylori. H. pylori is the causative agent of stomach ulcers, or at least a large number of them. So that's the idea behind pleomorphic. Want to recognize what that term means. So next we'll get to what are called arrangements. So arrangements are the ways in which bacteria group together. So you have individual cell shapes, and then you have multiple cells grouping together, and we refer to that as arrangement. Could have called it grouping, but they chose the word arrangement, so that's what we have to all be familiar with. Okay, there's a lot of that in science. If I haven't said already, the namings, I'm not a big fan. I'm really not, but it is unfortunate that that's the way it is, and we have to be, we just have to conform to these terms. So arrangement really just means grouping, and when we look at how bacteria often associate, we see that many of them kind of have these patterns by which they cluster together. And so that's a term that comes from a very common observation, that they tend to group. They don't tend to always grow separately. Some do, but they a lot of times tend to grow in this type of a certain arrangement. So when we look at these, we go back to the three most common shapes: cocci, bacilli, and then the spirillum. And then we look then at how the arrangements occur within each of those cell shapes themselves. Okay, so we have the three most common shapes and then the common arrangements within those shapes. Now let me just jump to the end here to kind of cross this one off. Spiral shaped bacteria, the spirillum, and the spirillum and the spirochetes, they typically do not form groups. They are most commonly in a what's called a singular arrangement, which is really the only one that implies a lack of grouping. Perhaps that's why they don't call it grouping, because there are some that don't group. They're actually considered a singular. So when it comes to the spiral shape, that is almost always what you find there. There are always exceptions to that, but generally speaking, that's the norm there. So we can just think of that as just kind of being those that lack any sort of grouping. Now, when it comes to the cocci, the circular shaped bacteria, they tend to form five. Really, there's another one here that I'll show you that isn't on this list. I want you to add that one, so I'll pull that one up here in a second. But you have what's singular, meaning that they don't group. Diplococci means two. Diplo means two, so you have two together. Staphylococci, staph means cluster. I'll write that out here in just a second. Strepto means chain, so you have chains of circular bacteria, so you have clusters of them, chains of them, and then tetra means four, so tetrads, you have a group of four here. So let me pull up the write the prefixes here. Staph, let's see, I think staph by itself refers loosely to some sort of cluster, and strepto refers to a chain. So when we see a staphylococci, this is in reference to cocci bacteria in clusters. All right, we can say the same thing here about the streptococci, except rather they are cocci bacteria in chains. Okay, here's the pictures for those. So singular, diplo, staphylo, strepto, and then tetrads. Now the one I want you to add, let me pull that one up real quick. I should have this ready, sorry. Here it is. There's one more that's commonly seen called a sarcina. Sarcina is like a tetrad, except there are kind of like two tetrads. So it's 8 in this kind of cuboidal type shape, as opposed to only the 4 in a tetrad shape there. In fact, actually I do have that one on here, I'm sorry. Here's another view of it. Sarcina. I was going to show that next, but I'll show it now. Okay. That's the one I didn't have on here. So those are the common arrangements in the cocci. Those are the ones I want you to know. And again, typically if I give you a figure, it's going to look more like this, so it's clear as to what we're talking about. Bacilli. The bacilli tend to occur in about 4 common grouping patterns. Again, like the cocci, they can be singular, so lacking a grouping. They can be diplo, like the cocci. They can be strepto, like the cocci, so diplo bacilli, strepto bacilli. They don't have a staphylo bacilli. Apparently, that doesn't happen, so you don't have that as a common grouping because it doesn't exist. So you just have to recognize there is no staphylo bacilli. And then the last one that is unique to bacilli are called palisades. And palisades occur where bacteria kind of grow next to each other. Let's see if we can find a better figure here. I always like to show as many different examples as I can. Here's one showing palisades. These are computer animations. This is a actual image here. It's a little harder to tell when you put it under a microscope. Mother Nature is never quite as simple as the textbook image, but that's kind of the idea. They grow next to each other. Here's a figure showing how they grow next to each other. So that's the idea behind palisades. Okay, So once again, how they group together and common examples of that. You do want to be familiar with the spelling as well as how to identify examples. And just to clarify one more time, I'm not going to try and trick you. If I give you images, I'll make sure that they don't have really kind of subjective looks or subjective answers. Like, for example, before I talk about this, I would not take this picture and ask you to differentiate between these two. I can't even tell the difference there. I cannot. You can kind of tell if that's Sarkanae, but it's relatively difficult to tell those two apart. I wouldn't ask you to do that, okay? But I might. This one's fairly obvious that it's a chain of cocci, so might give you that picture or something like that and ask you to tell me what that is. But when it comes to these types, I'm not going to try and trick you on that. So just know the basic stuff and don't worry about any sort of trickery. I don't like to do that. So, but just to clarify, I do want you to know the terms, the spelling, and I want you to be able to identify examples of those. Okay, so next, I also want you to be familiar with just basics here of what determines arrangement without getting into all the nuts and bolts and details. From a kind of a general perspective, why do they have these arrangements in the first place? Why do some have a diplococci and some have a strepto and some have a staphylo? What's going on there? Two kind of general things. One is what's referred to as a pattern of division, or also called a plane of division. And that is basically the point in a cell where the two cells will split apart during cell reproduction. So one of the things that happens when bacteria are growing is that they divide and make copies of themselves. So one cell will literally split apart to form two cells, right? And hopefully you're kind of familiar with that general premise. When they do this, they typically will have a point at which the one cell begins to split apart. So you can see that here. Here's one cell, and they've shown it right here by this plane division where that cell would start to split apart to become two cells. So the exact location of that is the first part of that. Now, one of the things that happens is that in some bacteria, the plane of division is always in the same spot. So there's kind of this marker where it always is in the same location. In other bacteria, the plane of division can rotate, sometimes in a kind of an even interval. So in a tetrad, what's actually happening is that the plane of division starts in one location, and then it rotates, and then it rotates again. In a Sarkanae, what happens is that same thing, but then it rotates on its axis to rotate into more of a horizontal axis as opposed to a vertical axis. So you're getting multiple rotations of that particular plane of division. That's how you can get these cells in different layers and in different angles. And then in Staphylococci, it's not showing it here, but the plane of division is random. Every time it goes in a different angle, it can be a different horizontal or vertical axis. And as a result, the growth is somewhat random, but in a clustered type form. So that is what is partially explaining that. The other real basic aspect is the attachment. Why do, or I should say, when do they separate? The why part is actually a little bit harder to explain, and I honestly couldn't tell you every detail of that. But what is happening is sometimes they split apart and sometimes they don't, and that's the basic answer there. Why that is, honestly, I don't honestly know. I don't know why sometimes they form two and then the rest of them split apart from there. Honestly, I couldn't tell you. I'm sure somebody has a basic answer on that, but those are things that are actually generally harder to answer, why that is as opposed to what's happening. So I can tell you what's happening. The plane of division is sometimes altering, rotating, and then the attachment after the division. Sometimes they stay attached, sometimes they break apart. So based on the nature of those two things, you'll get different types of arrangements.